friends. Okay. Again, there's other chemical requirements, phosphorus, sulfur, some trace elements, all of those are in your medium. Not to be confused with, this is what makes the microbes happy. We have not talked about selective media yet. We will get to that. This is just, hey, a little extra nitrogen makes this microbe a little happier. Not, without it, it won't grow, and then you can determine what it is by that. There's two different things. We're just talking about what microbes like. Right? Like what they like to eat, what makes them happy. Here's some other growth factors, amino acids, obviously cholesterol, heme, NADH, an electron carrier, niacin, PABA, some different things that are in different nutrient medias. Okay, physical requirements. We will talk about this. Also, as you know, most of the bacteria you're using are grown in at 35 or 37 degrees. We can change the temperature a little bit, and you all have a little incubator there. The temperature tends to affect the three-dimensional structure of proteins. Yes, remember DNA makes amino acids make protein, and the way they fold causes them to make correct or incorrect proteins. So protein three-dimensional structure is pretty important. Lipid-containing membranes are temperature sensitive. If the temperature is too low, the membranes become too fragile. If the temperature is too high, it sort of melts the membranes and they become too fluid. So we don't really want either of those things to happen. Here is some growth charts. So this is like minimum, so it'll grow maybe at room temperature, maybe it'll even grow in the refrigerator. Typically when we take your stuff out of the growth incubation, we put it in a refrigerator to keep it from minimum growth. You come up here like this and you get a increase in growth rate, and then you do hit a peak. This optimum is usually like 30 degrees, 37 degrees. It can get hotter and then it just dies at a certain point. So I would say this optimum is probably 37 degrees. Here's 30 degrees. You can't see a huge difference, but there's just a little bit better growth at 37 degrees. You can sometimes use this as a selective media. It'll be like it won't grow at 37 degrees, or it won't grow until you get to 37 degrees. It won't grow at 22 degrees. So sometimes you can put your plates in various conditions and, delegate between two. But mostly this information is given to you so that you know how best to grow your bacteria. Okay, and then of course we have extremes. We have psychrophils. These guys grow like in the tundra, in the freezing cold water. And then you have hyperthermophils or thermophiles. Thermophiles, usually these grow in hot springs. So then you have like sort of normal temperature, and then you have really cold and really hot. So here's some growing. Look at the ice growing in extremely cold, like negative 30 degree temperatures. Physical requirements. So pH obviously is going to be something necessary when you're thinking of what kind of media you want to grow. The effects of water. Once your media dries up, like if you leave your agar out too long, it'll dry up and your bacteria will die because it won't have anything to live on, and it especially needs water. Microbes require water because it dissolves enzymes and nutrients, and it's important as a reactant in many metabolic reactions, and water is just pretty important. Most cells die in the absence of water. Some have cell walls that retain water better than others. And sometimes when you remove the water, they make endospores. So, and cysts will cease their microbial activity, but they will like curl up into an endospore without water. Two physical effects of water that we need to talk about in relation to bacteria. We have osmotic pressure. Do you remember osmotic pressure? This is the cells expand or contract based on where water is. So if there's a higher concentration in something, if there's a higher concentration inside my semi-permeable cup, the water will want to rush into the cup to dilute it. Water likes to dilute things. If there's water on the outside and there's a higher concentration, the water will come from my cup to the water outside. If this was a semi-permeable membrane. It's not, but if it was, that's what would be happening. If this was a blood cell or something, water would be rushing in to dilute it, or water would be coming out to dilute it, or it would stay the same. And hydrostatic pressure is the pressure either on the cell or pushing the other way from the cell. So these are both really important in terms of the growth of bacteria. So osmotic pressure, again, is related to water is going to rush in to dilute things. Water's job is to dilute things. So here you have a semi-permeable membrane, and water will rush in and change this pressure, this osmotic pressure. It's exerted on a semi-permeable membrane by solution containing solutes that cannot freely cross the membrane. So here's the pressure, right? There's osmotic pressure going this way, and there's back pressure going this way. Your capillaries, there's blood flow, there's hydrostatic pressure going out, osmotic pressure going in. That keeps things from imploding. Same with your blood cells. What do you want to have? You want to have hypertonic blood cells. Yeah, you do. Do you want to have hyper? No, you do not. So basically, this is water rushing out of the blood cell to dilute the outside, right? This is not good. You do not want your blood cells all shriveled up. This means you are hypertonic to your environment and water's coming out. Isotonic is what you want. Look at those nice little guys, right? There's water coming in, water coming out. Everybody's happy. Hypotonic. This is blood cells bursting. Anybody think that's bad? Raise your hand. Yeah, I don't want my blood cells to burst. What's happening here? There's a higher concentration inside than outside. This is when people put things in DI water, right? You got a goldfish at a fair, you throw it in some DI water, you don't let it acclimate, its little cells are going to burst, right? You give someone IV of straight water, their blood cells are going to burst, right? This is not good. So these are the solutions. Obligate and facultative sometimes are restrict the solution on the outside because otherwise they have this effect, which we don't want. Well, unless we do, right? That's the weird thing about microbiology. In my mind, I'm like, yeah, we want them to die. We want to kill them all. Like, I don't want E. coli. Nobody wants E. coli. But for the purposes of this lab, we're trying to keep the E. coli alive. And the more we know about how to keep it alive, perhaps the better we will be at killing it. Okay, so hydrostatic pressure. You have barophils, and these guys live under insanely high pressure. It's the only way they can live. Now, we don't have any of these in the lab, obviously, because we don't have a way to create that kind of pressure. What's the difference between halophils and barophils? They are extremophiles, right? They live in extreme conditions. Halophils require a saline environment, so they have to have 9% NaCl, kind of like your blood cells. And barophils require extreme hydrostatic pressure. So those are some examples. Why do we care about this? We care about this because we need to know how these things grow. We need to know why they grow in these crazy environments. And we need to know all this information so we can kill them. There are some that we want, but we also need to know this because we need to know how to identify them, right? So you do a simple stain, you know if you have bacilli or cocci, you do another gram stain. Then you look at like, will it grow in mannitol salt? Or what color does it turn? MacConkey agar. Does it grow in MacConkey agar? Does it grow in a certain stain? So there are some of these that we use to look at dichotomous keys. I don't know if this is going to work, but we shall try. So this is your whole end of your project.
Let's practice using a key to identify this unknown sample. Whether the unknown has gram-positive cells.
A gram stain of this unknown shows red, gram-negative cells. Thus, the answer is no. The key then tells us to go to step 3. Also from the gram stain, we can tell that the cells are indeed rod-shaped or bacilli. The key then tells us to go to step 4. Only bacteria that tolerate oxygen will grow in a medium exposed to air. This growth is visible as cloudiness or turbidity. As you can see, our unknown became turbid in a medium exposed to air. The key then tells us to go to step 5. Lactose fermentation can be determined by growing bacteria in a medium that contains lactose and phenyl red.